Today I will talk with you about the functional role of an Alzheimer's disease-associated poly-T site in the TOM40 gene. So my lab research program, the main goal is to find out the significant and the functional consequences of genomic region or gene that were found to be associated with neurodegenerative disease of the aging population in general. Our starting point is from genes that have been identified through a genomics association study, candidate-based or whole genome association study. And we then follow up with these genes by pinpointing the actual causal variant and the molecular mechanism of action. And we're using several approaches, bioinformatic tool, human brain tissues, and ex vivo and in vivo model system. Our ultimate goals are to identify novel therapeutic targets and diagnostic biomarker. We're working primarily on 3 genome regions. Region in the genome, alpha-synuclein in relation to Parkinson's disease and Lewy body-related diseases, SORL1 in relation to Alzheimer's disease, and the TOM40 ApoE lot score genomic region in relation to Alzheimer's and cognitive decline in aging. And today I will talk about the TOM40 ApoE genomic region in relation to Alzheimer's disease. So our overall hypothesis is that changes in the expression level of normal protein in the brain can lead to neurodegenerative diseases. Regulation of gene expression has different levels: genetics, epigenomics, environmental. Within environmental, we can talk about exposure to pesticides, for example, diet, and other factors that were mentioned early this morning. Another factor for gene regulation is aging and gender. I will focus today on the genetics, and specifically our interest is in non-coding structural variants. We think that they are the main player in gene regulation in the brain that lead to diseases. And today I will talk about a specific example. So ApoE lot score region and AD, the relation between them, the association, have been known for a while. Back at 1993, the ApoE4 allele of the apoprotein E gene was the first genetic risk factor to be identified for the non-familial late-onset Alzheimer's disease. And ever since then, it's remained the most reproducible with the largest effect size of AD genetics. It was shown that carrier of the E4 allele have earlier age of onset. Later on, when data from genome-wide association study emerged, and all the integrated and meta-analysis show that the ApoE linkage disequilibrium region is by wide margin more significantly associated with increased risk for Alzheimer's disease, late-onset Alzheimer's disease. And I listed here a few of the whole genome-wide association studies that started to be reported and published through, at 2009. But what was interesting from the observation of these genome-wide association studies are, first, when you look at all the signals, the significant signals across the genomes that found association with Alzheimer's disease, the ApoE linkage disequilibrium region was most significantly, but not in few fold, by wide margin, but by a very high magnitude comparing to the other signals for association. And another interesting point was that the marker across this region were mapped within neighboring genes. So what we ask ourselves is, only the ApoE epsilon 4 can explain this high significantly association signals, or there is something else there? So this is the linkage disequilibrium region of ApoE on chromosome 19, and we can see that we have here the ApoE gene and the TOM40 upstream, PVL are upstream from the TOM40, and also ApoE-C. So some of the marker for the association, this region, were mapped to the 5' region of TOM40 and also for ApoE-C. I'm talking about the genome-wide association studies. So we looked, we magnified into this genomic region, and by deep sequencing analysis of fuzz sequencing in fuzz, intact chromosomes, followed by haplotype analysis and phylogenetics analysis, we identified here a poly-T variant that was associated with LOD. So let me just tell you a little bit about this poly-T variant. There are 3 groups of alleles. The short, which are the poly-T sequence is 19 and less. The middle, between 20 to 29 T's residues, and the very long, which are longer than 30 T's in consequences. We characterize this allele by range and by frequency of the groups in different populations from different ethnicities. And you can see that the range is very wide in people from like African American and people from Ghana, from the African origin, and also that the distribution, the shorter allele is much more frequent within this population comparing to Caucasian. And this is a summary of the different population, the characteristic of this element, sequence element in the genome. So we also look at the haplotype pattern between the ApoE epsilon, the E2, 3, and 4, and this poly-T, and this is summarized in this graph. And our conclusion from the haplotype structure is that in white and Hispanic, the middle allele, the L allele, is primarily linked to ApoE4, while the majority of the VL and the S alleles are linked to ApoE3. In contrast, in African American, Ghanaian, and Japanese, there is an increased frequency of the haplotype that link the short poly-T with epsilon, with ApoE epsilon for haplotype. We look at the association between late onset Alzheimer's disease to this poly-T site and did some survival curves and show that the VL allele, the very long poly-T stretch, are associated with early disease onset, earlier than the short. This was done in a population that are compound heterozygote to epsilon 3 and 4. And this was the original study by Rosas et al. That was published in 2009. Later on, another group studied subjects that are homozygote for epsilon 3 and replicate this study. But I have to stop here and say there are other studies with conflicting results, and this is a contemporary topic. We pursue and look at the genetics of cognitive changing in elderly, normal elderly, and we wanted to see if this polity contributes also to cognitive performance in normal aging. So we started an outreach project in the local retirement community of independent living, people that are cognitively healthy. We collected personal demographic details, lifestyle, cognitive performance using different, wide battery of neuropsychological testing. And we collected also biological samples. We collected saliva for DNA and blood from which we extracted RNA and a protein. So this is the demographic, the descriptions of our study population. They are all from the Triangle area at North Carolina. We characterize here, we give the characterization by age, gender, their education, all the data that we collected by questionnaires, the data that we collected by performing neuropsychiatric testing, and our genotyping data. I will focus a little bit more on the genotyping data. These are the allele frequency and the genotype frequency for both ApoE genotypes and the poly-T element in TOM40. They are linked, they are close by, as I showed before in the genomic structure. We perform association between the poly-T site to our cognitive data, the comprehensive cognitive test, individual test. And we restricted the analysis to subjects that are ApoE33 in order to look independently on the effect of this TOM40 poly-T site. And we showed that the homozygote for the shorter allele performs significantly better than the other genotype group on measure of specific cognitive domain of memory and executive control and executive function that are preferably show deterioration in early stage of Alzheimer's disease. We also look at the expression levels in their blood cells of genes within these clusters in relation to cognitive performance. And there are trends towards significance in the relation of expression of both ApoE messenger RNA and ApoC messenger RNA with specific cognitive testing. So what we know about this poly-T is that it's associated with risk for late onset Alzheimer's disease. It's associated with age of onset of late Alzheimer's disease. It shows also association with cognitive performance in normal aging. And there are other groups that shows also association between this poly-T site and biomarker in the CSF of Alzheimer's disease. So now what we were interested in is to study the molecular mechanism of action, what actually this poly-T does. So our hypothesis was that the TOM40 poly-T has a regulatory function, and it's modulate the expression, regional modulator of the gene within the ApoE TOM40 region, LD region. And thereby its impact on the pathway in which this protein participates and mediate the late onset Alzheimer's pathogenesis. So how do we study this? There was few support in the literature that poly-T site or this specific poly-T site can act as a regulator of gene expression. There are other examples in vitro study that shows that poly-T tracks can act as an enhancer element for regulating transcription via their effect on nucleosome organization. There is a group that shows that an extended region upstream of ApoE and including TOM40 gene has an effect on gene expression of ApoE in brain and CSF. And there is also an in vitro study in cell-based systems that show that small regions that flank of this poly-T within reporter system in cell-based, in cell culture, can regulate expression of the luciferase genes. So what we proceed to do. So we did some messenger RNA analysis using human brain tissue, and we focused on occipital cortex and temporal cortex. And this is an overview of our brain study, a sample. We have here the number of temporal cortex and occipital cortex. Most of them are matched samples from the same subject. We took samples from a patient, like autopsy. All of these samples, of course, are autopsy, and a normal. And I added here some of the demographic. We can see that the post-mortem interval are low, so our RNA integrity was very high. And we focus on Caucasian only. And in this specific study, in the first step of this specific study, we looked at subjects that are ApoE33, homozygote, because we want to see the independent effect of this poly-T. So first, we compare between control, normal by a clinical and post-mortem examination, and we see that TOM40 messenger RNA is increased in both temporal and occipital cortex in AD patient compared to control. When we look at the TOM40 messenger RNA expression in relation to the poly-T variant, in normal brain, we can see that the VL homozygote, we have like an increased level in both brain structure when we look at the normal brain. When we look at the AD brain association, and just pay attention that everything here is the ApoE33, we observe exactly the same relation, but we've reached high significance with the VL allele associated with increased levels of TOM messenger RNA. So now we look at ApoE, the neighboring gene, the adjacent gene. Here, the association, the relation in expression were even more standing out. So again, we did exactly the same analysis. Messenger RNA of ApoE were increased in highly significant between in AD patient compared to control in both brain structures. Looking at AD disease brain, we can see significant association between the poly-T genotypes to expression level of ApoE messenger RNA, when the VL again lead to higher expression. And with the normal, we see trends toward the same direction, but they were not significant for ApoE. Now we broadened our study samples and included also So before we're moving on, so this shows that we have an association between the poly-T independently of the ApoE genotype and expression of messenger RNA of both Tom40 and ApoE. So now we extended and we included in the analysis also ApoE 3-4. First, we just wanted to look between the expression levels of the genes Tom40 and ApoE. To compare the homozygote to the compound heterozygote. So we see the homozygote for the 3-3 have higher expression level compared to the 3-4, which probably indicate that the pathological effect of ApoE4 is on the protein level. It's related to the functional or dominant function of the protein that is translated from the E4 allele, while the E3 effect probably working in the transcriptional level. Okay, when we look at the 3-4 only, we got the same trend. Heterozygotes for the VL show higher expression of both Tom40 and ApoE compared to heterozygote to the LLL, but it didn't reach significant. The last thing that we did, we combined the both study population and compare subjects that carry the short allele comparing to subjects that carry the very long allele. And again, we see the same trends. The VL lead to higher expression level in both brain structure and both genes, Tom40 and ApoE. Okay, so we see that when we're taking the intact brain tissue, which contain heterogeneous cell types, glia, like microglia, astrocytes, neurons, we have an effect on expression. But what really interests us, and what I think that will let us know which pathway are involved in the pathogenesis of AD, is to pinpoint which cell type is actually responsible for the expression change. So what we're currently doing, and I don't have final result, I just want to present the approach, is we're doing expression analysis on homogeneous pool of cells that we're isolating only neuron, only astrocyte, and only microglia, about 200 from each cell type. And we're doing it using the laser capture microdissection, what we call LCM. We're starting with frozen brain tissue from disease and healthy. We cutting under chill, cool conditions. This slide, we're staining with markers that specifically stain each of the cell line. And then using the LCM apparatus, we're isolating specifically neurons, astrocyte, and microglia. Then we extract the RNA, we amplify the RNA because the number of cells per pool is 200, so we need to amplify that. And we're using the encounter single cell expression assay from nanostring to measure the expression of genes in the ApoE Tom40 genomic region. So this is ongoing study. I hope that next time I can tell you the results from this approach. We took another approach to validate or to confirm the effect of the poly-T on the expression of gene. We used the luciferase reporter assay with different 5'3" poly-T alleles, but we didn't use only the poly-T and short flanking site. We took the whole genomic region from the translational start of ApoE throughout intron 6 of Tom40 that include the 5'3", so we can imitate the genetic or the genomic context, the real genomic context. And we confirm in both HepG2, which is a liver originated cell line, and SHSY5Y, which is a neuroblastoma cell line, that the VL allele led to higher luciferase expression, which basically stands for a gene within this genomic region. So here we go. We have also a cell-based system to validate our result from human brain. And currently we are in the final stage in making humanized mice by replacement with bugs that contain 40 kb from the human, from PVLR2, which is 3 prime, through ApoC1. We're replacing the mouse genomic region with the human genomic region, and we're looking forward to measure the gene expression in the mouse brain, and then do some functional and mitochondrial functional study with these mice. So to summarize, TOM40 is associated with a late onset Alzheimer's disease, age of onset and risk. This is the first study that basically drive all of our functional study. Expression of gene at this genomic region, this locus, is associated with AD status, disease, yes or no. TOM40, the poly-T, is associated with cognitive performance in normal aging. And also we show association trend of this gene locus expression in blood with cognitive performance. So these genes play a role, their level play a role in cognitive performance in aging. Expression of gene in the TOM40 ApoE locus in healthy and diseased brain is associated with these poly-T genotypes. So just to conclude, the poly-T act as a regional regulator of TOM40 and ApoE gene expression of the messenger RNA on the transcriptional level, and this probably the molecular mechanisms that can explain the genetics association of this TOM40 poly-T with AD and cognition. And I would like to thank my lab teams and our collaborator at Duke Neurology. Thank you.